Hello everyone, I'm Professor Lee of Dr. Anti-Aging and Herbal Medicine TV. This is the third lecture on aging treatment by removing senescent cells. Today, we'll learn about various diagnostic methods of senescent cells accumulated in our body, methods for preventing senescent cells, and lifestyle habits that have been proven to be effective in removing senescent cells. Let's get started. If senescent cells are the major cause of accelerated aging and disease occurrence, will removing senescent cells slow down aging, prolong life, and improve disease? The answer to this question is yes at present. It's been proven effective in animal studies, and the results of early clinical studies are also very positive. Now, let's look at the supporting evidence. Among drugs that kill senescent cells are called senescent cell removers, or Senolytics in English. The first senolytic pill was a combination of dasatinib and quercetin. Dasatinib was originally an anti-cancer drug, but it also promotes the death of senescent cells. Quercetin is a natural flavonoid found in large quantities in onions, apple peels, broccoli, strawberries, and grapes, and it helps to remove senescent cells along with the antioxidant effect. Mice that had their senescent cells removed by the combination of dasatinib and quercetin lived 6 months longer, which is equivalent to extending their lifespan by 5 to 10 years in humans. Inflammatory substances level were also reduced by 30 to 60%. Importantly, mice that had about one-third of their senescent cells removed continued to have effect even with just one administration. In another study, middle-aged mice were given the drug orally once every 3 weeks for 3 months, and the rate of aging was also significantly slowed. The result of these animal studies showed that removing senescent cells could not only prevent disease progression, but fundamentally slow down the aging process. And importantly, after removing senescent cells, not only did they live longer, but they also lived longer and healthier lives. These mice suffered less from age-related disease and remained energetic until their final moments of death. There are also reports that removing senescent cells not only slows down the aging process, but actually makes body functions younger. In 2016, mice that had their senescent cells removed with dasatinib and quercetin had stronger muscles, moved more actively, and performed new exercise tasks much better, just like younger mice. In another study, after removing senescent cells, the mice's blood vessels became more flexible, their blood pressure was lowered, and their risk of cardiovascular disease was reduced. In other animal studies, skin wounds healed much faster, and the regenerative capacity of major organs, such as the liver and lungs, was significantly increased. Diabetes, osteoporosis, lung disease, and dementia have also improved. Now, I'll tell you about the result of a clinical study. The first small-scale preliminary clinical study was conducted on patients with idiopathic pulmonary fibrosis. Idiopathic pulmonary fibrosis is a chronic disease in which lung tissue thickens and hardens and lung function continues to decline. When senescent cells accumulate in the lung tissue, chronic inflammation continues, hardening the lung tissue, causing shortness of breath even with a little movement. 14 patients with idiopathic pulmonary fibrosis were administered 100 mg of dasatinib per day and 1,250 mg of quercetin per day, 3 times a week for 3 weeks. Physical functions such as the 6-minute walking distance, 4-meter walking speed, and chair rise time significantly improved. As for side effects, one of the 14 patients reported a major side effect, and other minor side effects were respiratory symptoms, skin irritation and bruising, and digestive discomfort. This first study provides initial evidence that senolytic pills can improve physical functions in some disease. Just as the field of cancer research has made tremendous progress and achieved many results thanks to the concentration of enormous research funds, a lot of research funds are currently being supported for research related to senescent cells. Therefore, It's important to note that Senolytics, which is a senescent cell removal agent, are expected to be the first to be prescribed as an anti-aging treatment in clinical practice due to the tremendous speed of research development. I'll explain in detail about the numerous recent clinical studies and various senolytic pills in Part 4. In order to apply senescent cell removal in clinical practice, there must be a diagnostic method, right? I'll now explain the main diagnostic method for finding senescent cell problems in our body. The first method is to stain senescent cells and make them appear blue. Tissues collected or cultured cells are used. Since many waste products accumulate inside senescent cells, they secrete a lot of an enzyme called β-galactosidase to break them down. As you can see, the granules containing this enzyme increase inside the senescent cells. This enzyme turns blue when it reacts with a specific substance, so you can observe it with your eyes. We are seeing now testicular cells of a mouse, and you can quickly tell whether these cells are senescent cells. After staining like this, almost all of them are stained blue, right? Almost all of them are senescent cells. The second method is to measure inflammatory substances secreted by senescent cells. It's easy to test with just blood, and if a lot of these substances are found in the blood, it's a sign that there are a lot of senescent cells. A 2022 study analyzed levels of senescent cell secretion in the blood of people with high levels of senescent cells to predict the progression of chronic disease. The method could be particularly useful in managing disease associated with old age. Thirdly, if you want to stop the growth of damaged cells, you should naturally send a signal to stop, right? If you measure the protein that acts as this stop signal in the tissue or blood, and the level is high, it means that there are many senescent cells. This test method is used in research to find people who age quickly, or in clinical studies to observe changes after senolytic administration. In addition, there are also analyses of function of proteolytic enzymes that increase in senescent cells, DNA damage analysis, and genetic analysis. In the future, we'll combine these tests with MRI and PET scan tests to directly observe the effect of senescent cell removal treatment. From now on, I'll tell you about the lifestyle method that have been proven to prevent senescent cells. The main causes of senescent cells are DNA damage and telomere shortening, so it's no different from methods to prevent DNA damage and telomere shortening. However, among the various methods, weight management is particularly important in preventing senescent cells. Overweight and obesity are closely related to chronic inflammation, and senescent cell accumulation is also closely related to chronic inflammation. Therefore, if an overweight person also has the problem of senescent cell accumulation, aging progresses very quickly. In fact, in one clinical study, the level of cell growth arrested signal protein, which increases when senescent cells accumulate, was 40% higher in the overweight group than in the normal weight group. Weight management plays a really important role in preventing senescent cell accumulation. GLP-1 hormone proliferation, which are hot worldwide these days for diabetes and obesity treatment, can also be viewed as anti-aging treatment from the perspective of senescent cells. In addition to weight management, other important preventive measures are the same as those mentioned in the DNA and telomere sections. Aerobic exercise 3 to 5 times a week, 30 to 60 minutes per session, prevents cells from transitioning to a senescent state. After about 3 months of regular aerobic exercise, the level of growth arrested signal protein that increases in senescent cell immune cells decreased by 30%. This is because the senescent cell of an immune cell was significantly slowed. It also reduced the number of senescent cells in the thigh fat tissue of overweight elderly women. In elderly people who exercise regularly, vascular endothelial cell senescence in the brachial artery was also reduced. Sufficient sleep of 7 to 8 hours a day reduces chronic inflammation and prevent the accumulation of senescent cells. In particular, in elderly people, even if sleep deprivation lasts only 1 day, the level of protein and inflammatory substances that increase in senescent cells in the blood increases. women with long-term insomnia have more senescent immune cells in their blood than women who get enough sleep. I want to emphasize again that sleep is not just a matter of fatigue, but it's also very important in preventing aging. Chronic stress increases senescent cells and inflammation levels. Studies show that if chronic stress is not effectively managed, senescent cell-related indicators and inflammatory levels can increase by more than 20%. Stress management techniques such as meditation and yoga can help alleviate this. From now on, I'll tell you about lifestyle habits that have a direct effect on removing senescent cells, not just preventive effect. The first is calorie restriction, which is effective not only for removing senescent cells, but also for everything related to aging, such as extending lifespan. The calorie restriction diet is a diet that reduces calories intake by 10 to 25% while maintaining balanced nutrient. It has been proven in animals for a long time that it delays aging and extends lifespan, and also been shown to reduce the number of senescent cells in humans. The second, intermittent fasting, is also a very important anti-aging longevity method. There is the 5-2 intermittent fasting method, which involves eating normally for 5 days a week and restricting calories for 2 days, and the 16-8 intermittent fasting method, which involves the fasting for 16 hours a day and eating for the remaining 8 hours. The specific practice method will be explained in detail in the autophagy chapter. Third, high-intensity interval training has a remarkable effect in removing senescent cells. For example, cycling or running is a type of exercise that involves exercising for 1 minute to the point of being out of breath, followed by a 2-minute recovery period, and repeating this for at least 3 to 10 times. In a study of 39 adults aged 18 to 60, after 3 months of high-intensity interval training, the amount of protein that increased in senescent cells in muscle cells decreased by 35%, and the level of inflammation also decreased by 25%. In a 2024 study that observed the elderly for 12 years, the level of 10 or so proteins that increased in senescent cells decreased. I'll explain more about high-intensity interval training in the mitochondria chapter. That's all for today's lecture. There are now 4 and 5 parts left in the senescent cell removal lecture. In the next 4th part, we'll study the best food, nutrient, and herbs that have been proven to prevent or remove senescent cells. And in the last lecture, 5th part, we'll study various senescent cell removal pills. Thank you all for your hard work, and I'll see you again next time.